Hello, my name is Andrey Fedorov at Brigham and Women's Hospital. I'm one of the leads of the Imaging Data Commons. And in this presentation, I want you to give an introduction into the capabilities of this new resource of the NCI Data Commons. I'm presenting this on behalf of the team. IDC is developed by a consortium of partners, which includes Mass General Brigham, with Ron Kikinis serving as the PI of the project. Bill Longabo at the Institute of Systems Biology is leading the cloud architecture of the resource. David Pott is our lead in security. David Clooney is leading data modeling efforts. Steve Piper and Rob Lewis are the experts in setting up the visualization capabilities of IDC. And Hugo Ertz and Andrey Fedorov are the leads in developing use cases in radiomics and pathomics that will be utilizing Imaging Data Commons for cancer research. Imaging Data Commons is a new component of the broader network, NCI Cancer Research Data Commons, or CRDC, which aims to bring together data, computational capabilities to the cloud to support cancer research, sharing of the data, and development and validation of new tools. You can learn more about Cancer Research Data Commons at this URL. Imaging Data Commons is a new node of the Cancer Research Data Commons, which joins other data nodes, which include Genomics Data Commons, Proteomics Data Commons, and Canine Data Commons. The goal of Imaging Data Commons is to meet the needs of cancer imaging researchers in connecting researchers with the cancer imaging collections, as well as robust infrastructure that allows to explore, identify relevant data, visualize relevant data, and collocate that data with the computational capabilities that will allow to analyze it. In addition, Imaging Data Commons aims to bring imaging data and link that data with the other types of modalities and resources to support cross-omics analysis. It is important to emphasize that Imaging Data Commons is just one of the components of CRDC, and other components such as NCI Cloud resources and various other tools are intended to connect together the data and computational capabilities and support deployment of workflows on the cloud. The main function of the Imaging Data Commons is to support cloud-based analysis. We want to make sure that the cloud can serve the needs of cancer imaging research. We want to allow you to build the cohorts focused on imaging and ultimately support integration of imaging with other types of data. In order to allow meaningful research and meaningful sharing of data and analysis results, we aim to achieve fair science and follow the FAIR principles of data provenance. Our goal is to establish the standard of how to use standardization and harmonization of data to facilitate discovery and curation of metadata. And we want to support these principles by the best practices worked out examples. Development of use cases is part of our plan. And as I mentioned before, our team includes experts that will be developing use cases that utilize the platform we are developing. All of the components we are developing will be shared as open source, and we truly believe that individual components that we utilize in IDC will also be helpful and useful as standalone tools and will be useful to others. Finally, but very importantly, we want to enable community building. We want to establish an open forum that will include cancer researchers, clinicians, scientists, and also non-imaging researchers to understand what is the best way to use cloud to advance cancer imaging research and discovery of new treatments. IDC contract was awarded last year, in the summer of last year. In October, we had the pilot release of the platform, and the capabilities of the pilot IDC includes support of radiology data and hosts a subset of the data collections, public data collections from the Cancer Imaging Archive. We plan to have production release of IDC next year, and the extended capabilities of the resource will include, in addition to all of the radiology collections from TCIA, also capabilities to visualize digital pathology data. And we are also actively working with other projects, such as Human Tumor Atlas Network, to extend the availability of data available within IDC. You can access the Imaging Data Commons at this URL, portal.imaging.datacommons.cancer.gov. Here is the idea for the overall workflow of how we believe user would interact with the Imaging Data Commons. The idea is that without having to install any software, just using the browser, the web browser, the user would be able to identify and browse relevant imaging collections, identify subsets of this data based on the rich metadata, do the quality check and visualization of the selected images from that cohort to develop confidence in the quality of data. And once that is done, use off-the-shelf analytics tools, and I will give you some examples of what those tools could be, to further do refinement of the cohort, do some exploratory analysis, further search using the metadata. And once the cohort is finalized, the user can use customized cloud-based virtual machines to perform the analysis, and once the workflows for the analysis are set up on a single VM, scale it up to use multiple machines and apply the analysis on a large amount of data. Finally, we want to enable the researchers to share analysis results and workflows and to achieve more reproducible science and improved reproducibility of the research. What I want to do next is to show you the capabilities of the portal, which right now includes 27 collections from TCIA, and this includes both the original imaging collections and some of the collections that include analysis results. So here you see the main page for the portal. You can see a quick summary of the data which is included in the portal. You can also see specific collections from TCIA that are currently available within IDC. If you click on exploration, this will bring you to the main cohort builder interface. On the left, you will see various facets, various attributes of metadata that we expose. And this includes both the original attributes that describe imaging collections, but also derived data. As you modify the selection on the left, you will see that the visualizations on the right update dynamically. As for example, here you will see various types of annotations that are available for the chest imaging collections. You're also able to subset your data based on the attributes of the derived data, such as selecting all studies that include segmentation of a lung nodule, for example, and you can, when available, you can filter the subset further by the size of the nodule. You are also able to visualize individual studies or series in the IDC viewer, and IDC viewer that is included allows you to see not just the images, but also the annotations available in those images. A few more details about the viewer that we provide. We utilize open source Open Health Imaging Foundation viewer, and every time you open an image in IDC viewer, it will use the DICOM web interface to pull the data from the DICOM stores that we maintain, and you can use both in-slice visualization using this viewer and also multi-plane reconstruction. You can also, of course, in MPR, you can see the annotations. In terms of annotations, we support DICOM segmentation objects as well as radiotherapy structure sets, as I show here in this example. We also support layouts. So, for example, you can see multiple series at the same time. Here, the visualization of PET images is shown side by side with CT images and the overlay segmentation results. However, those capabilities that I showed you is just scratching the surface of what you can do with IDC. Very importantly, IDC maintains not just the tools to explore the data, but also the actual data. And our two main venues for working with data, first is storage buckets that contain all of the DICOM files that we indexed in the resource. You can use those buckets to pull the data to a virtual machine, and that is going to be very fast and free as long as your VM is co-located in the same region as the data. And we also provide BigQuery tables that incorporate DICOM metadata that we search, and also those attributes that are not exposed in the cohort builder of IDC. So you can use this to further refine your cohort and explore the content of the data that IDC is hosting. Here is one example of what you can do with the BigQuery tables. Here, what I'm showing you is using Google Data Studio, which is a free tool that you can use to build dashboards that interface with large data tables, such as in this example, it's working with the BigQuery tables that IDC is maintained. You can see that here we integrate the interface to the viewer with the dashboard. So as you refine the definition of the cohort you are building, you can look at the individual results. And you can very quickly add new attributes to your dashboard. For example, such attribute as the manufacturer as shown here. And you can very quickly filter your data to, for example, see a subset of data for just a single manufacturer. So this gives you more flexibility of how you can explore the data and find data that is relevant to you. And you can build this kind of dashboard in a matter of minutes. Another interface that is again available is Google Colab. Google Colab or Google Collaboratory is the hosted instance of Jupyter Notebook. Google again will provide this to you for free and will allocate a virtual machine, and you can even allocate a virtual machine that has a GPU to run your notebook, to pull the data from the buckets to this virtual machine. You can also use Colab notebook interface to interface with the BigQuery tables. So in this example, we explore the attributes of LADC collection. And for example, here we show how variable is the slice thickness for this collection. And again, the viewer can be easily integrated with the Colab notebook, and you can readily visualize specific images that you selected using the Colab interface. Now, of course, you can do a lot with Google Colab, but there's a limit to what you can do with free resources. Once you know how to do the analysis, once you're confident in your cohort composition, you can use Google Compute or Google AI notebooks to configure the hardware and software environment. And you can replicate this at a number of virtual machines, including programmatically, and run your analysis on large amounts of data using highly scalable Google Compute Platform infrastructure. In order to achieve all those capabilities that I showed you, there are several key components that we utilize. First is the data representation. We rely on the DICOM standard to represent both the images and the analysis results, and we actively work on harmonizing data that is not in DICOM into this representation to support our data modeling efforts and search and query interface. There are many collections in TCIA that utilize DICOM for the representation of analysis results, and all of those collections will be available within IDC. To support visualization, we rely on Open Health Imaging Foundation Viewer. This is a viewer developed to a large degree supported by NIH funding, and it's a very flexible platform used both in academic and commercial projects. It supports DICOM web interface that we utilize to pull the data into the browser. It's a zero footprint viewer, means the data is downloaded into your browser before it can be rendered. We already have the experimental results using OHAF for pathology data, and that's one of the directions of our current effort. In the past year, as we were working on the development of the IDC pilot, we worked very closely with the OHAF team, and actually OHAF team is part of IDC team. All of the contributions that we implemented in OHAF are released as open source and are available to the broader community for reuse. The foundation of IDC is Google Cloud Platform. Google Cloud Platform provides a number of tools to support storage, exploration, and analysis of data at scale. And Google Cloud Platform also implements a Google Cloud Healthcare API, which includes implementation of DICOM standard, which is very critical for our purposes. In the past year, we worked a lot on the development of our procedures, how to utilize the cloud, how to organize the data, how to establish the security procedures, and also worked very hard on interfacing with TCIA to pull the data using TCIA API to IDC. As I mentioned, the data on IDC comes from TCIA, and TCIA and IDC are partners in bringing fair data to cancer imaging researchers. However, there are certain roles that TCIA plays and is different from IDC. TCIA supports curation and identification of data, and these tasks are not handled by IDC at this point. The goal of IDC is to support cloud-based hosting of data co-located with the compute resources. We also actively work on harmonization of data into DICOM format, such as our efforts to harmonize annotations and digital pathology data into DICOM format. And importantly, IDC will be hosting only public collections, while TCIA supports hosting and download of private and public collections. And again, if you want to download the data from IDC to your local machine, this will be quite expensive, while downloading data from IDC to a virtual machine on Google Cloud will be free. If you want to download it to your laptop, TCIA is the tool of choice for you. IDC is one of the components of CRDC. Other components include proteomics, genomics, and canine data commons. And it's important for the imaging researchers to be aware of those resources. Cancer data aggregator is a component of CRDC, which is coming online next year, that will provide API to support aggregated queries and exploration of data across different nodes of CRDC. This resource will be very important for enabling cross-omics analysis and cross-node data exploration. Finally, I want to mention that in addition to the technical capabilities of the platform, we also bring the documentation. We will have the ability to award cloud credits for the early adopters to explore the capabilities of the resource. We also have a forum for our users that we hope you will use to ask your questions and engage in discussions with developers and fellow users of the platform, because your feedback is invaluable for us as we refine the capabilities of IDC and decide what should be the next steps and priorities for us. In the coming year, our priorities include refinement of our extract, transform, load processes, refinement of API, collaboration with TCIA, development of use cases. We are really looking forward to get your feedback about what you want to see and what capabilities you need to have in IDC and CRDC. As I mentioned, we work towards digital pathology visualization support in IDC, but also we plan to have all of the public radiology TCIA collections by the spring next year available in IDC. If you want to learn more about Imaging Data Commons, here are the two URLs that you can use, and we really look forward to interaction with you and meeting you at our user forum. Thank you.